Welcome to this deep dive by Biotechnology Guy. And just a heads up for everyone listening, this is going to be a very detailed one today. We are really getting into the weeds of some heavy astrobiology.
Yeah, we're definitely going to be unpacking a lot of complex chemistry, but, the goal is to explain all the jargon so it makes sense for the non-biotech folks out there, too.
Right, exactly. We want to make this an engaging science education type of journey for you because, we're tackling essentially the biggest existential question we have. Like, where did life actually come from?
And we are not talking philosophically here.
No, chemically. Like, before there were cells, before there was any kind of metabolism, where did the specific molecules that can store biological information actually come from?
It's basically the ultimate locked room mystery,
Mhm.
except the room is the entire early solar system, and the lock was sealed 4.5 billion years ago.
Right. And today's source material, which is just a phenomenal breakdown provided by Biotechnology Guy, it looks at a landmark study published in Nature Astronomy. This came out on March 16th, 2026.
Yeah, and the hook here is just wild, because this study, well, it didn't definitively answer the origin of life completely, but it brought us closer than we have ever been.
And they did it using a sample of dust that weighs less than a teaspoon of sugar.
Much less, actually.
Yeah, less than a teaspoon of sugar collected from a rock drifting 300 million kilometers away from Earth.
It's just staggering when you really think about the logistics of it.
It is. But to really understand why this tiny pinch of dust is so revolutionary, we have to talk about the rock it came from and the completely wild heist of a mission it took to get it.
Absolutely. The target was an asteroid named Ryugu.
Right. So for everyone listening, let's break down the setting here. Ryugu is about 900 meters wide. It's orbiting the sun between Earth and Mars, and it's shaped kind of like a spinning top. But the most important thing is that it is a C-type asteroid.
Right. And we should definitely bust some jargon here. So C-type stands for carbonaceous.
Which means it's carbon-rich, right?
Exactly. It's carbon-rich, it's ancient, and it is chemically primitive.
Okay, so it's not just like a floating lump of coal out there.
No, not at all. When we say primitive, we mean it's essentially a rubble pile made of the leftover building blocks from the very formation of the solar system, 4.6 billion years ago.
So it's basically a chemical time capsule.
That is the perfect analogy, yeah. Because it's just been sitting out in the cold vacuum of space, it preserves the exact conditions of the early solar system. It completely bypassed the extreme heating and melting that happened on planets like Earth.
Right, because when Earth formed, it melted down, heavy stuff sank to the core, lighter stuff floated up, and all that original chemistry was totally destroyed.
Completely erased.
But
Ryugu is like this pristine, frozen snapshot.
which is why the Japanese Aerospace Exploration Agency, or JAXA, decided they absolutely needed a piece of it. And the Hayabusa 2 mission is just, the logistics are staggering.
Oh, it's an engineering marvel.
Right. It launched in 2014, traveled for 4 years to get there in 2018, and then didn't drop the sample back to Earth until 2020. 6 years of flight for literally 5.4 grams of rock.
And getting that 5.4 grams was incredibly dangerous. Because when they finally arrived at Ryugu, the surface was not what they expected at all.
Yeah, they thought it would be somewhat smooth, right? Like powdery plains.
Yeah, they were hoping for a nice, safe, dusty landing site. But when the high-resolution cameras turned on, they just saw this treacherous nightmare of jagged, monolithic boulders.
Boulders everywhere.
Everywhere. And you have to remember, a spacecraft is fragile. It's got these big, delicate solar panels spreading out. If you land on a boulder, the mission is instantly over.
So they had a huge problem.
A massive problem. They had to delay everything for months just to map the surface down to the centimeter to find a flat clearing.
And the clearing they found was what, like a target circle, just a couple of meters wide?
Barely. And the landing technique they used to hit that target is just incredible. Ryugu is tumbling, it's spinning, and the communication delay with Earth is over 15 minutes.
So you can't joystick it. You can't have a guy with a controller adjusting the descent.
No, it has to be autonomous. They achieved a pinpoint landing accuracy of 60 centimeters on a spinning rock 300 million kilometers away.
That is just sci-fi levels of precision. But the actual collection wasn't like a little robot arm scooping up dirt, right?
No, because in microgravity, if you try to press a shovel into the ground, the force just pushes the spacecraft back up into space.
Right. Equal and opposite reactions. So what did they do?
They used this meter-long cylindrical horn that stuck out from the bottom of the spacecraft. The instant the tip of that horn touched the asteroid, it fired a tantalum bullet.
Wait, a literal bullet?
A literal bullet. A 5-gram projectile fired at 300 meters per second directly into the rock.
That is so aggressive.
It had to be. The kinetic energy shattered the surface, and all that dust and debris kicked up through the horn and into a collection chamber.
Okay, so that gets them the surface material, but the surface has been getting blasted by the sun for billions of years, right?
Exactly. Solar radiation, cosmic rays, micrometeorites. It alters the organic chemistry. So, the surface sample is great, but the subsurface material is the holy grail.
Right, because that stuff is totally shielded. So how did they get underneath the surface? I know they used the small carry-on impactor experiment.
Yeah, they basically used explosives. They hovered above the asteroid and dropped this device packed with high explosives, backed by a copper plate.
Like an anti-tank weapon?
Basically, yeah, a shaped charge.
Yeah.
They retreated behind the asteroid for safety, detonated it, and drove that copper plate into the surface at 2 kilometers per second.
Wow. And that made an artificial crater.
It blew a 10-meter crater right into Ryugu, exposing material that hadn't seen the light of day since the solar system formed. Then they went back down and did the bullet trick a second time inside the crater.
That is just phenomenal. But, getting the sample was really only half the battle. Because the reason this specific 5.4 grams is the most important rock in science right now isn't just because it's from Ryugu, it's how they brought it home.
The contamination issue. This is everything.
Right. So let's talk about the historical problem here. Because finding organic molecules in space rocks isn't technically a new thing. We've had the Murchison meteorite that fell in Australia back in 1969, and the Orgueil meteorite in France way back in 1864.
Yeah, scientists have been finding amino acids and organics in those meteorites for a long time.
But there's this huge asterisk, right? The asterisk problem. Because Murchison landed on a literal sheep farm. Orgueil was sitting in rural France.
They were exposed to the Earth.
Exactly. They sat on the ground in the berg getting rain on. Humans picked them up with bare hands. So whenever scientists claim they found space organics in those rocks in the past, were they just measuring Earth dirt, like sheep droppings?
Well, I wouldn't say it was just sheep droppings. Scientists use isotopic ratios to feel confident that the organics are extraterrestrial. But you are absolutely right that absolute certainty was impossible. Contamination is a ghost that haunts meteorite analysis. If a soil bacteria gets into a porous space rock, it ruins the data.
Right. There was always that lingering doubt. But Hayabusa 2 completely fixed that.
They engineered an impenetrable seal. The return capsule came down, fiery re-entry, parachuted into the South Australian outback, and the recovery team had it flown to Japan in just 57 hours.
And the room they opened it in was basically cleaner than a surgical operating theater.
Oh, way cleaner. It was a Class 100 clean room.
Right, which is the standard they use for manufacturing spacecraft components and semiconductors. It's completely free of microorganisms.
And they didn't even open it in Earth air. The sample was pressurized and opened in ultra-pure nitrogen gas.
Right, so there's no oxygen to degrade the molecules, no moisture, no microbes, just pure nitrogen. The takeaway for you listening is the absolute certainty this provides. What they find in this dust is from Ryugu. There is zero Earth contamination.
Zero, which is why the global scientific community practically begged for a piece of this 5.4 grams.
Yeah, I bet. But finding the actual molecules of life in there required a really delicate touch. Which brings us to the next phase of the story, the initial incomplete picture.
Right. So the samples arrived in late 2020. And by 2023, the first wave of research came out. They found some incredible stuff, including uracil, which is a major nucleobase, and niacin, which is vitamin B3.
But that was kind of a problem, right? Because they found uracil, but the other 4 nucleobases that make up our DNA and RNA were completely missing.
Exactly. And people were confused. Was Ryugu just weirdly specialized to only make uracil?
Enter Toshiki Koga from Jamstech.
Yes, the Japan Agency for Marine Earth Science and Technology. Koga had a hunch. He didn't think the other molecules were missing at all. He thought the sample sizes they were analyzing were just too small for the older detection methods to spot them.
So he applies for more dust, and JXA gives him 20 milligrams.
20 milligrams. It is an unfathomably small amount.
Like less than a grain of rice, right?
Yeah, a single grain of long green rice is maybe 20 to 30 milligrams. This is like trying to find a needle in a haystack, but the haystack is the size of a tic-tac.
So we really need to bust some jargon here on how they actually analyze something that small. How do you do chemistry at the absolute frontier like this?
It requires an incredibly aggressive yet delicate extraction methodology.
Okay, so translate this for us. They start with water, right?
Yeah, they use a hot water extraction first to pull out any free-floating organic molecules, but that only gets the easy stuff. To really get into the mineral matrix, they use a 6-molar hydrochloric acid hydrolysis.
Which sounds intense. I mean, hydrochloric acid, you're basically dissolving the rock.
You're chemically teasing out every molecule that might be bound to larger organic polymers. The acid breaks those bonds and frees the nucleobases.
Okay, so now you have this tiny vial of highly acidic liquid soup filled with thousands of random space molecules. How do you find the specific letters of DNA in that mess?
That is where they use high-performance liquid chromatography, or HPLC.
Right, okay, let me try an analogy here. Imagine you have a massive bucket of mixed coins, and you pour them down a series of chutes at really high pressure.
Okay, I follow.
And the chutes are lined with, like, chemical Velcro. Different coins, or in this case molecules, interact with the Velcro differently. Some slide right past, and some get caught and slowed down. So they all travel at different speeds and emerge from the chute at very specific, predictable times. Does that work?
That's a great analogy for how the chromatography column works, yeah. You separate the mixture based on how highly polar the molecules are, so you can isolate them by time. But then you have to identify them.
Right, which brings in the high-resolution mass spectrometry.
Yes. Think of this as an impossibly precise scale.
Like how precise?
We are talking about measuring the exact mass of a single compound down to the 4th or 5th decimal place of an atomic mass unit.
Oh, wow.
It charges the molecules, accelerates them through magnetic field, and based on how they deflect, it weighs them, so we can identify them among parts per billion concentrations in that rock dust.
So you know exactly when the molecule exits the Velcro hallway, and you know exactly how much it weighs, down to a fraction of a decimal.
Exactly. And when Koga's team ran the 20 milligrams through this gauntlet, the result was unambiguous.
They found them.
They found them. All 5 canonical nucleobases: adenine, guanine, cytosine, thymine, and uracil.
In both the surface and subsurface samples?
Yes. Perfectly reproducible.
I mean, that is just a dramatic revelation. But to really appreciate why finding those 5 specific molecules is such a big deal, we need to pause and look at what they actually do. For you, for me, for every living thing on Earth.
We need to look at the alphabet of life.
Right. So let's do a quick tag team breakdown of genetic biology. What actually are nucleobases? Let's start with DNA.
Okay, DNA. Deoxyribonucleic acid, the famous double helix. The rungs of that twisting ladder are made of these nucleobases, and there are strict pairing rules.
Right, they only fit together in specific ways.
Exactly. Adenine, which we just call A, always pairs with thymine, or T, and guanine, G, always pairs with cytosine, C.
And that complementary pairing is the whole secret, right? That's what allows the DNA to be unzipped, copied, and repaired with perfect fidelity. If A only pairs with T, you always know what the other side of the ladder is supposed to look like.
Precisely. It is a brilliant, structurally perfect storage system.
And then there's RNA, ribonucleic acid, which is kind of like the working copy of the gene.
Yeah, it's what your cells use to take the instructions from the DNA and actually go build proteins.
And RNA uses the same letters, except it swaps out thymine for uracil, or U.
Right. So between DNA and RNA, you have these 5 canonical bases: A, G, C, T, and U.
And the thing I really want to emphasize here is that this is a language analogy, but it's not a loose analogy. These 5 molecules are the literal alphabet of life.
They are not optional components.
Right. Every single protein in every organism, every human, every blade of grass, every dinosaur that has ever lived was built using combinations of these 5 exact letters. If you remove even one of them, the entire biological system crashes.
It just doesn't work. The geometry of the hydrogen bonds fails.
Which brings up the massive historical astrobiology debate. Because for decades, the standard assumption was that these letters were invented here on Earth.
Yes, going all the way back to the 1953 Urey-Miller experiment.
Where they zapped the gases with electricity, right?
Exactly. The idea was that the early Earth was covered in a primordial soup of hydrogen cyanide, ammonia, and water. And then lightning strikes, or sheet from hydrothermal vents, forced these simple molecules to assemble into the complex nucleobases.
But the Ryugu discovery forces a total paradigm shift, because now we have this alternative theory that they didn't form here, they were delivered from space.
The exogenous delivery theory, during the late heavy bombardment billions of years ago.
And finding them in Ryugu makes this delivery theory incredibly concrete. These molecular prerequisites for life are not unique to Earth at all. They're just natural products of the solar system's chemical evolution.
They were forged in a vacuum before Earth even had liquid oceans.
Which is mind-blowing on its own. But as biotechnology guy points out in the analysis, finding the 5 letters was just the headline, because buried in this paper is this weird chemical ratio that hints at a completely unknown space laboratory.
Yes, the data on the purines and pyrimidines. This is where it gets deeply fascinating.
Okay, so jargon buster time again. What is the difference between a purine and a pyrimidine?
It's just their physical structure. Adenine and guanine are purines, meaning they have a double ring chemical structure. Cytosine, thymine, and uracil are pyrimidines, meaning they have a smaller single ring structure.
Okay, double ring purines, single ring pyrimidines. Got it. And what the team found was this finding within the finding. The ratio of purines to pyrimidines actually correlates negatively with how much ammonia is in the asteroid.
Which is something nobody expected to see.
Right, let's look at the data across the different rocks. So Ryugu has an intermediate, kind of middle-of-the-road ammonia level, and its purine to pyrimidine ratio is like 1.1 to 1.2, so nearly equal amounts of both.
Right. But then you look at other meteorites. The Bennu and Urgile meteorites have much more ammonia in them, and they lean heavily toward the single-ring pyrimidines.
And then the Murchison meteorite has less ammonia, and it leans heavily toward the double-ring purines. So there is a clear sliding scale linked directly to ammonia. Why is that so mind-blowing?
Because no known formation mechanism predicts this relationship. We have models for how these molecules might form prebiotically, but nothing in our current chemistry textbooks explains why ammonia would dictate this exact ratio in a space rock.
So you're saying nature found a way to mass-produce DNA letters in space that human science hasn't even figured out yet?
Exactly. It points to a completely undiscovered chemical pathway operating in the early solar system. We haven't been able to reproduce it in Earth labs.
That is just wild. And to add to that, the team didn't just find the canonical 5 letters, they found non-canonical compounds too, right? Things like hypoxanthine and xanthine.
Yes, those are purine intermediate.
And 6-methyluracil, which is a structural cousin to thymine. So, I mean, let's use a factory analogy. It's like we aren't just finding the finished, polished letters rolling off the assembly line. We're finding the scrap metal, the half-built chassis, the rough drafts.
That's a brilliant way to look at it. It proves that Ryugu wasn't just accidentally spilling out nucleobases randomly.
Right.
It was running an active, systematic biochemical pathway. It's a precursor chemical environment that actually mirrors how living cells build and metabolize nucleotides today.
It's like the asteroid was practicing biology before biology existed.
Essentially, yes.
Yeah.
In a purely chemical sense.
But if this massive, organized chemical factory was happening inside Ryugu, was it happening everywhere? I mean, Ryugu is just one rock.
Well, this is the beauty of modern space exploration. Another mission gave us the answer to that exact question.
NASA's OSIRIS-REx mission.
Yes. While Japan was visiting Ryugu, NASA went to the C-type asteroid Bennu. They collected samples and brought them back to Earth in September of 2023.
And the data from that mission just came out, right?
Yes. A companion paper was published in Communications Chemistry in April 2026, and they confirmed the exact same thing.
They found the whole toolkit.
The complete toolkit. All 5 nucleobases are present in Bennu as well.
So just to summarize the sheer magnitude of this for everyone, we have 2 different missions conducted by 2 different space agencies, going to 2 completely different ancient asteroids, and they come back with the exact same message.
The rock chemistry of life is relatively common. It's everywhere in these ancient rocks.
Okay, but I need to explicitly stop the hype train right here, because we all know how the internet works. People hear this and immediately think, did we find life on an asteroid?
And we have to emphatically clarify what Koga said about this. The answer is no.
Absolute no.
Ryugu is a cold, dead vacuum. There is no liquid water currently. There is no energy gradient to power metabolism. There are no cell membranes to hold things together.
Right. Finding the alphabet does not mean you found a book.
Exactly. Nucleobases are necessary for life, but they are far from sufficient. You need so much more to actually spark biology.
But what it does mean for the early Earth is incredible. If we picture the young Earth, say 4.5 to 3.8 billion years ago, it's a hellscape, but it has oceans. It has heat, and it is enduring the late heavy bombardment.
Right, millions of asteroids crashing down.
So if these carbonaceous asteroids contained all 5 nucleobases 4.6 billion years ago, then for hundreds of millions of years, it was literally raining the alphabet of life onto the young Earth.
continuously raining down. Trillions of tons of material, the meteors break up in the atmosphere, and the dust settles.
And those molecules dissolve right into the primordial oceans. They wash into the hydrothermal pools.
Which completely shifts the fundamental question of astrobiology.
Right, because for decades we were asking, how did the Earth manage to magically synthesize these incredibly complex nucleobases in a hostile atmosphere? And now we know it probably didn't have to.
We no longer need to ask how they formed on Earth. We now must ask, what happened to the nucleobases once they were delivered, fully formed, from space?
How did the Earth take those pre-made letters and stitch them together into the first RNA strand? That is the new frontier.
And it is a much more focused, answerable question now that the bottleneck of making the bases themselves has been solved by the solar system.
So just to recap this incredible journey for you listening, we started with a 900-meter tumbling rock out in the void. We executed a pinpoint 60-centimeter landing. We fired a bullet, blew a crater, and sealed the dust away from the Earth.
Brought it to a flawless clean room in Japan.
And from just 20 milligrams of pristine dust, science extracted the fundamental alphabet of biology.
It's a deeply humbling reality, because the letters that encode your DNA, every living thing on this planet, they were forged in a cold, radiation-bathed vacuum millions of miles away.
Before the Earth even had oceans.
It didn't take a warm, wet planet to make the ingredients for life. It just took a carbon-rich asteroid, a lot of time, and the chemistry of the early solar system.
Which leaves us with a final, really mind-expanding concept to chew on as we wrap up this deep dive. Because if the universe appears to have been perfectly manufacturing the alphabet of life for as long as there have been rocks to make it in,
And we know these rocks are everywhere.
right, they're everywhere. The alphabet is universal. So what exactly did it take to assemble those letters into something that could read itself?
Yeah.
Something that could copy itself, evolve, and eventually launch a spaceship to go retrieve its own chemical ancestors? Because if the alphabet is everywhere, so where are all the other books?
That is the big question.
It really is. Thank you all for joining us on this special deep dive. Keep questioning the chemistry, and we will see you next time.